Hi, this is Dr. G. Bailey, and in this video, we're going to focus on topic 10.1, which is all about meiosis. It is an HL course. So here are some of the things that we're going to look on. We're going to look how chromosomes are replicating during before meiosis through the interphase. We're going to talk about crossover more in detail, how it is between non-sister homologous chromatids. We're going to talk about the new combination of alleles as you do the crossovers. We're going to talk about the chiasma formation between the non-sister chromatids. We're going to talk about homologous chromosomes separating during meiosis 1. We're going to talk about the independent assortment of genes due to random orientation of the homologous chromosomes. We're going to talk about sister chromatids separating meiosis 2. Also, we're going to draw a diagram of the chiasmata that's formed by crossover. So here I'm going to do a quick overview of meiosis, although this is something that you already learned in topic 3. It's just a quick recap. So the whole thing is we have a homologous pair of chromosomes in which you have the parents here going to duplicate. So that means you can see there's a duplicate for each one of these chromatids. So when you have a duplicate, they become known as sister chromatids. And then once you have them going through this, obviously these guys are going to separate eventually into each one of them being in one of the gametes. So this is what we know, that in the end, we're starting with a diploid, and in the end, we're ending with 4 haploid daughter cells. That's what's the whole process of meiosis, because meiosis is about producing, as we know, sperms or eggs, right? And the girl will be eggs and the man will be sperms, and each one of them will have the half, which is usually 23. So basically we start with 46, but we end with 23. So meiosis is all about the reduction. That means we're reducing the numbers of chromosomes in these sperms and eggs. Now we're going to talk a little bit more about homologous pair and sister chromatids so you can really understand what the wording of these. But before we do, first let's understand how we get to sister chromatids. This usually starts all the way in the interphase. In the interphase, you usually have that one chromosome right here, right? Which it doesn't have its second copy. So the replication is happening during the interphase, in which we're duplicating the chromosomes, and that usually happens during the S phase. If you remember, there's G1, S, G2, it happens in the S phase. Now, when this happens, you usually have a copy. And that copy is called the sister chromatids, in which they're held together by the centromeres. So you can see here there's made of 1, and then when you make a copy, now you have 2 of exactly the same thing. And right here in the middle, as you can see, that's where the centromere is holding those 2 chromatids together. So here I'm going to show you a concept. Just to imagine that we are actually made of 46 chromosomes, right? And when we go through the S phase, we duplicate them, right? And that means we end up with 92. Now, what exactly these 92 are? They are known as chromatid, as you can say. Now, these chromatids, basically, if we put it in other words, you can say that they are made of 46 pairs of chromatids. Now, when we say this, or we can say sister chromatids, students get lost with these words. Oh, 92 chromatids, or 46 pairs of 23 pairs of sister chromatids, or we say 46 in this case. So students get lost between the words and numbers. So when they get lost with this, they usually feel like, oh, I don't know how to play with the numbers. So I'm going to go from the most basic thing. If I'm letting you know if it's 23 or if it's 46, what exactly the numbers? So what I'm going to show you is the most step-by-step, so you finally can sink in the numbers and what's happening. So we know for sure that the inner cell is made of 46 cells, right? 46 chromosomes. So I'm going to put here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46. So here we have 46 chromosomes. This is a normal cell. Now it decided to go towards S phase, in which you're making its duplicates. So in duplicates, I'm going to put here with the red color. So I'm making a copy of it. So right here, what I just created, I created a copy. So this is known as a sister. That's a sister chromatid. That means these two are exactly the same. So I'm doing sister chromatids for all of the chromosomes in the cell. So I'm copying every single one of those 46 chromosomes. So when I'm copying every single one of these chromosomes, they are creating their sister, and their sister, when they are together with their sisters, they're known as sister chromatids. So if I come and ask you the question again, now I ask you, how many chromatids are here? Right? You will say, oh, there's 46 chromosomes, but there's a doubled, right? There's double of them now, so obviously there is 92 chromatids. Now it makes more sense. Now, if I ask you, how many sister chromatids are here? Now, if you look at this, how many there is? You go, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46. So we got what? 46 sister chromatids. So it's not 23. This is where students get the mistake. So there is 46 chromatids, there is 92 chromatids. But then comes the next question. How many homologous chromosomes there is, right? Now the homologous chromosomes is usually a pair of 4. So it will be, in this case, you'll be connecting, let me put another color, it's these, you're gonna have these two sisters, like they're completely different from each other. But as a homologous, this is a homologous. It's made of 4. So this is another 4, which again, each one of these homologous, we can also call them tetrads or bivalents. So we have 4, 4, 4, 4, 4. So you are composed of 2 sister chromatids, right? Another 4, another 4, another 4, another 4, another 4, another 4, another 4. So each of these are known as homologous chromosomes, right? So now you can see, oh, homologous chromosome is made of 2 sister chromatids. Now if I ask you how many homologous chromosomes are here, of course after the S phase, you can even count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. So we have what? 23 homologous chromosomes, or we can say homologous chromosomes pair, right? So there's 23 homologous chromosomes, there is 46 sister chromatids, there is 92 chromatids. That's how you will count these numbers. So right now, it makes more simple, it's way more easy to understand the concepts. Another thing that we need to understand is later, how does the homologous chromosomes pair works? So when we talk about homologous pair, I'm going to add here a little page here just to show you. So imagine you have here a sister, just one chromosome, which is made of A, B, and C. And then I go through duplication, right? The S phase, which then I make a sister. If I make a sister, it will look like A, B, and C. They have to look exactly. So these two are known as sister chromatids, right? And then you have another one. You're going to have to get a homologous. When you say homologous, is not the same sisters, okay? These are sisters chromatid. But they're not, these together, these are not sister of all of these. These all together is known as homologous. Now, when you say homologous chromosome, what is that makes them homologous? Because the word homologous means the same. What's the same is that here they have to have a gene that is A, B, and C. But they're not sisters, so that means this might be little a, little b, little b, little c, little c. Now, what they have in common, if you can see, they have the common is that they have the order of A, B, C. The genes are in the same location, the A's, the B's, and the C's. What's the difference between them? It is the difference between the alleles. These are big A's and these are small A's. So what makes them different? It's different alleles, even though they have the same what? Genes. Now, when do you do the crossover? Now, the crossover always happens right here between the non-sisters, which makes sense, right? Because this right here is not a sister of this, so they are not sisters, remember. It's completely different sisters. So that's where the crossover happens. Now, crossover can never happen between sisters. Why? Because you're just flipping a C with a C. It's going to be the same thing. Or a B with a B here. It's the same thing. So you'd never do a crossover between sister. The crossover will happen between the non-sisters. which is right here between these guys. So this is a way to clear up when we talk about crossover between the non-sisters from the homologous chromosomes. So as we had here, we have a sister and a sister connected by their centromeres, and when you put them together, this is known as a tetrads. Now this is where the synapses happen. This is basically where they're connecting the two sisters together. Now, this is where we go to the next part, which is talking about the crossover. Now, we know that the homologous chromosomes, also known as tetrads, cross over each other, and pieces of the chromosomes of genes are exchanged. So you can see here, right, there's an exchange. And when this exchange happens, you see that new pieces have been flipped between the two. So these are known as recombinations of the offspring. So this offspring, look, it's completely different than the parents. So as this is different parents. Why? Because of this crossover. So the crossover is always exchanging or the alleles inside. So here's another picture. You have a duplicate. You have a sister chromatid with another sister. They do crossover right here. And you can see the crossover is happening between non-sisters. And now you have different types of mixtures of recombinants of cross chromosomes, right? So what does the crossover does? Multiply already have a huge number of different gametes types produced. So you're making so many different types of sperms and you're making so many different types of eggs because crossover mixes the genes. So right here, a homologous chromosome, bivalent or tetrad just means the homologous. Now right here, where there is a connection happening between the two chromosomes, that's where the chiasma happens. What is chiasma? It's where the crossover happens. So chiasma is the location of crossover. And when they cross over, you can see that pieces have been exchanged. So when you do, for instance, right here, this, and you connect with another chromosome, right here, there is no chiasma. You don't see any. However, if I do this again, and I cross here with another one, now I do see a chiasma. Chiasma is right here where there's a crossing going on between the non-sisters. Now this is known as a chiasma. So every time you see a matching going on, like a cross between two chromosomes, that's where the chiasma is happening. And it's the chiasma where the crossover is happening. And this is important that you know this. So here is just showing a diagram how the alleles are exchanged, right? You have sister chromatids, and you have another group of sister chromatids. And then the crossover is happening right here. When the crossover is happening here, you can see there is an exchange happening. So we have here normal, and we have here the normal one. So where do you usually have this? You can see the big A, big D here, and you can see little a, little d here. And what's happening in the middle, where the exchange is happening, is that big A is going to go with little d, and little a is going to go with the big D. And you can see that these two, the purple ones, these are the recombinants. They have been the ones that did the crossover, the exchange. And now you have, look, you have 4 different types of chromosomes. That means you're going to have 4 different types of sperms or eggs. So let's play here a little bit just to help you do. So imagine a test, you receive this. So first you have to do its sister, which is exactly the same thing. And then you're crossing over with another non-sister. So there has to be big A, big B, and big C, right? So the crossover, as you know, it's going to happen between these two. So you will have, again, big A, small a, big C. This, I'm cross over the A, so it'll be big A, small b, and small me. And here I'm going to have little a, big B, big C. And then I have big A, big B, big C. So you see that I have 4 different chromosomes only because it happened here. Another way that you might come up in exams is, let's say you have here the chromosomes, be connected by, the chromatids connected by the centromere, and you have here basically another one, and we have it here. And I'm just going to put here a little bit of this X here. So I might ask you how many chiasma or how many chromosomes are happening here. So we have 1 chiasma here, 2 chiasma. So there's 2 chiasma. That means there's 2 crossovers happening. What if I have another one here? And then we have here a crossover, and then we have this. Look, we have 1, 2, 3. So we have 3 chiasma this crossover. So this is something that you need to understand whenever you're doing the chromosomes and the chiasma. So moving on, now that you understand a little bit about crossover, about the chiasma, and everything related to exchange. Let's now compare mitosis from meiosis. So they both are going to start with the 46 chromosomes, and then eventually they're going to go through the S phase and have 92, right, chromatids, or we say chromosome if you wish, and then we're going to go through the process. Now, the process has prophase, metaphase, anaphase, as you can see, and telophase, and then cytokinesis, and boom, you have the daughters. So in mitosis, you're going to have them come up visible, and then you're going to see them line up. Who's lined up in the middle? The sister chromatids. And mitosis is always about the sisters. There's no homologous. At the end, you're going to produce two identical cells, which are 2N, and they're both 46. When it comes to meiosis, what's the difference? They do first have two stages, meiosis 1 and 2. So you have prophase 1, prophase 2, metaphase 1, metaphase 2, anaphase 1, 2, and so forth. So in prophase 1, you have here the homologous happening, which that means there is a crossover, which only happens at prophase 1. It does not happen in prophase 2. Then they line up in the middle in metaphase. Now here in metaphase, what's happening? It is the homologous that is coming into the middle, not like the sister chromatids. And then they go through the process of separation, and from the separation, eventually at the end of the whole meiosis, you're going to have 4 cells, but they're going to be N, which means each one of them are just going to have 23. So meiosis is about the reduction of the chromosomes. You're going from 46 to 23. So looking more closer at meiosis here, we have them at prophase. They are doing the homologous, they're doing the crossover, and then they line up the homologous in the middle. Then you separate the homologous, and then you get telophase, cytokinesis, and then you go through meiosis 2, which prophase, again, they become visible, but this time it's just the sisters, and then the sister lineups in the middle, the sisters splits, and then you have all the 4 cells with 23 reduct. Now, what's important? When you're doing meiosis 1, you always mention homologous is in the middle, homologous is splitting. When you're meiosis 2, you always mention sister chromatids, right? They are the ones in the middle. It is the sister chromatids that are separating. So you got to be very careful because there's a lot of multiple choice where they're trying to see if you remember the trick. Homologous chromosomes for meiosis 1 and sister chromatids when you're talking meiosis 2. So here it's comparing meiosis with mitosis. And if you see in this picture, you will notice that meiosis 2 is very common with mitosis because look, here is just the sisters and sisters. And in meiosis 1, meiosis 2, there isn't. And then here you got, again, let me do another color, makes more sisters are separating, and you have the sister separatings here. So they're more in common. However, if you look at meiosis 1, it's not sisters. Here is homologous, it's homologous separating. You still have the homologous. So if we had to say which one is more common between meiosis and mitosis, it is meiosis 2 that is more common with meiosis 1 because they're talking about sisters chromatids in the middle, sister chromatids separating. Also, in meiosis 1, in prophase, there's crossover. So you don't have crossover happening in prophase 2. There's none of it. So that's why meiosis 2 is more close to mitosis than meiosis 1. So here is the most important thing. There might be questions like B which ask you to compare, distinguish, contrast the whole thing between meiosis and mitosis. And this is a perfect chart with full summary of all the possible answers you can have for this question. So let's go through. The first one, there are 2 round divisions. The only one that has 2 rounds is meiosis. 4 total cells is only meiosis. Duplicate chromosomes separate during the process. Now they do duplicate both of them. DNA is copied during interphase. They both duplicate before they start. At the end, chromosome number is the same as the original. That's only in mitosis. So you start with 46, you end with 46. The process produces body cells. That's only mitosis. Body cell means heart cells, lung cells, skin cells. The process produces cells that are the same as the original one. That's only mitosis. The process produces cells that are different from the original one. That's meiosis because you're producing sperms and eggs, which are completely different because of crossover. The process produces sex cells, which again, it's meiosis. The process produces cells with half a number, that's meiosis, because you're going from 46 to now, which is only 23. The process that only one round of division is mitosis. Chromosome pairs separates during the process. When I say chromosomes pairs, now we're talking about the homologous chromosomes, which only happens in meiosis. Two total cells are produced. That's only mitosis. So this is a very good summary of the entire process between mitosis and meiosis. You know all these possibilities. You answer any questions related to it. So just to keep up with the rest of this, remember how the cell cycle had G1 going to S to G2, and then it had an option. So here in S, it goes from 46 to make a copy of 92. When it gets to G1, it has two options. It can go to meiosis or it can go to mitosis. The only difference is mitosis, this is any part of the body. When it comes to meiosis, this is only on the what we call as the reproductive organs. Oh, let me write this again. So meiosis is only when you have the reproductive organs like the testes and the ovaries. Now, mitosis, 46 starts, and you're going to end up having 46, two of them, right? Two. And meiosis here, you're going to go from 46 to become 23, and you're actually going to have four of them. Now, what is these? These are sperms and eggs. And why do you want to do that? Because you want the sperm eggs to be 23, so when an egg that is 23 joining with a sperm, they go back to being 46, which is a diploid, and then the baby will be born with every single cell of them being 46. So the whole point of meiosis is all about reduction. You want to reduce the chromosomes, while mitosis is only about identical, which means you want to repair a cell, you want to grow a cell, but that's pretty much it. So with that, we finished the topic 10.1. Here are the study guides. All these answers have been answered in this video. If you know how to answer them on your own, writing them down, then you're strong for this section. Thank you.